Any questions about any of the material that we're going to cover on the exam?
For the X-linked dominant pedigrees, do we always assume the affected is a heterozygous?
Okay.
Or homozygous?
For X-linked dominant pedigrees, let's see, which parent do you want to have affected in this case? Okay, so we've got an affected mom. It's X-linked dominant. So we're going to draw a few kids. So the question is this what's this mom's genotype? Is she heterozygous or is she homozygous for the dominant mutation? Okay, so if it looks like this, if you saw this and I told you it was X-linked dominant, what would you do? Choice A, freak out. Choice B, call me a liar. Right, so if I gave you the first generation, you had to fill in the second generation. Right, then, which I'm assuming this is coming from the study guide. Yeah, what do? You tell me. So you make the decision. If there's no evidence that you can draw on to tell you whether or not that individual is homozygous or heterozygous, you pick. You say, I assume, write it on the exam, I assume that this parent has this genotype, therefore I filled in the next generation the way I did. So if her pedigree looks like this, what's her genotype? Is she heterozygous or is she homozygous for the mutation? She's heterozygous. She's got one wild type copy, one mutant copy. So the kids that are affected by this dominant mutation got her mutant allele. The ones that are fine, unaffected, had her wild type allele. Okay. What if instead, well, you tell me, what does it look like if she's homozygous for the mutation? Every single kid gets the mutation because they have to get a chromosome from mom. Both of mom's chromosomes have the mutation on them. So if you have that second generation data, then you can figure out what mom's genotype was. Are half the kids affected by the dominant disease phenotype, or are all of them affected? That'll tell you what the genotype of the mom is. Follow-up questions or other questions?
So following up to that, so for X-linked dominant, it could be heterozygous or homozygous? Okay.
Right, because you don't, yeah, one copy or two copies of the mutation, you get the phenotype. So dominant diseases kind of suck in that sense for you as geneticists, not for the, well, also for the people that are affected by the disease, but it's hard to tell what the genotype is. How does this compare to a recessive mutation? If this pedigree was recessive, right, if somebody's affected, let me erase some of this first. If this was recessive, then you know for sure that mom's, if it's recessive, she has to have two mutations in order to have the phenotype. So then you know right away what her genotype is, regardless of having to look at the next generation. It's recessive. If you know that in advance, or you predict that it's recessive based on maybe the pedigree, the phenotype skips every generation, you say, I think this is recessive. If you assume it's recessive, you know the genotype. Let's assume that this is recessive, just for a practice question. So now assuming that we're talking about a recessive disease, and you see this inheritance pattern. Mom's affected and two of her offspring are affected. How would you explain that? somebody else, both parents have to have the mutation in order for some of their kids to inherit 2 copies of the mutation. So dad must be a carrier. He's unaffected, his symbol wasn't filled in, but he has to have 1 copy of that mutation. That's the only way that those kids that are affected could each inherit the mutation from both parents. I think I've scribbled on that slide long enough. Other questions? Yeah.
If it's X-linked, the father can ever be homozygous, right? Because the Y wouldn't.
Okay. So X-linked traits. Oh dear. No. X-linked traits. Can the dad be homozygous? Certainly not. Well, there's a very rare case that yes, but we won't talk about that rare case. So what would you call dad's genotype if we're talking about an X-linked trait? How many X chromosomes does he have? 1. Right, dad's X and Y. So when I write a genotype for a male, when I'm talking about the X chromosome and the Y chromosome, if it's an X-linked trait, I would write something like, if dad's affected, let's say. What's his genotype? How do you describe the genotype? Right, star over asterisk over a hyphen. The hyphen indicating that there is no chromosome there. That is, he doesn't have a second X chromosome. He's only got 1 X chromosome. So on his X chromosome, he has a mutation. On his Y chromosome, that gene doesn't exist. There's nothing there. So that's the symbology for a genotype for male. So getting back to the original question, can a male be homozygous for a mutation that's X-linked? No, because there's only 1 X chromosome. So we don't call that homozygous. Does anybody remember what term we use for that? Hemizygous. Half-zygous, because he's only got half of the chromosomes of a diploid for the X chromosome.
So if the disease is X-linked and dominant and the guy has it,
Yes.
so then dominant will be on the X, versus if it's a recessive disease, would the guy just be a carrier and not affected because there's no.
Okay, so if this is X-linked dominant, let's do that in blue. How does he pass this on to the next generation? Right, he passes that mutant X chromosome onto all of his daughters, by definition, because all of his sons inherit his Y chromosome. Okay, so if it's dominant, that means his daughters are affected. They got 1 copy of the mutation, sons not. How does this change if it's recessive? So the male has an X chromosome that has a recessive mutation. Right, so he still passes his X chromosome to all of his daughters, but now his daughters are not affected because it's not dominant. His daughters are simply carriers. And for sex-linked, well, you can do either one of 2 things. You can put the dot in the middle of the symbol, or you can fill the symbol half in. I'd be happy with either.
So would the dad be considered affected
Ah.
or a carrier?
So that's the question, right? That was your original question. What goes in here for an X-linked recessive trait? They are affected. So the key question then is, why are they affected? It's a recessive disease and the male only has one mutant copy of this gene. Why does he show the phenotype?
Because there's no dominant X.
Right. So I'm hearing a smattering of responses. If I put them all together, then what you said was the male only has one X chromosome. So he doesn't have the wild type version on his other chromosome, the Y. The Y chromosome doesn't have most of the genes that are on the X chromosome. That's why this homozygous notation is important. So he has that recessive mutation on his X chromosome, but there's nothing there on his Y chromosome to provide the wild type copy that would happen if you had an X-linked or an autosomal recessive condition. If you're a carrier, that's because you're not showing the phenotype because your wild type copy that's on the other chromosome substitutes. You still got one normal version of the gene. Males only, for X-linked traits, our Y chromosome helps us out not one bit. Okay. Totally pathetic, degenerated wasteland of a gene chromosome. Just, it makes us male and that's about it. So that's the special case of X-linked recessive disorders. If a male has one copy of the mutation, it's not dominant, it's still recessive, but one copy of the mutation in males gives you the disorder. So in this case, dad's affected and passes it to all of his daughters are carriers. Other questions? So there's the pedigree of this family from Central America who had autosomal dominant deafness. And I asked you which region of which chromosome is associated with the inheritance of this disease. And a lot of you submitted answers, so if, I'm just offering this as an option. If you want me to talk about this now, I will. Okay. So this is called haplotype mapping. I'm not sure if I actually mentioned that the first time we went through this. I'm not trying to introduce new material for the exam. It's just called haplotype mapping, but we've done this in the last few weeks. So what are we looking for? We have a bunch of people who have deafness, filled in symbols. Let's zoom in right there. What are each one of these sets of characters? D5S 210, D5S 207, D5S 119. We've got a chromosome here. So go ahead. They're genes. They're genetic markers. But we can just consider them to be genes. So specific sequences of DNA along a chromosome. So what do these sets of numbers represent? Every individual has 2 numbers for every gene. So this dead female was 6 and 5 associated with FIB5. So that could be, for example, I'm not saying this is true, she might have a CA dinucleotide repeat that has 6 units, CA, 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 CA. So we just reduce that allele to the number 6 copies of that dinucleotide. And that would mean that on the left side, one of her chromosomes, because she's diploid, her allele is CA6. On the other one, that 5 means that she's got a difference on that chromosome. She got 5 copies of CA in a row. So you've got genes going down the chromosome, and then the set of two numbers tells you the two genotypes or haplotypes, one inherited from each parent. So just from this window, what do all of the individuals that have deafness share that the individuals that don't share? Looking for a correlation between which versions of the genes they inherited and whether or not they also inherited deafness. So for example, what about here? Is the genotype at chromosome, by the way, that 5 means we're looking at chromosome 5. So on chromosome 5, at marker 3210, is there a correlation between the genotypes of the individuals and whether or not they got deafness? So this individual inherited 2 3s and is deaf. Does everybody else who's deaf have 1 or 2 allele 3s at this locus? No. So we've got one individual here, although what's missing? We don't know one of their haplotypes. So there might be a 3 in there, we don't know. They don't have that genetic data. Same for this individual. So we actually can't rule out at this point whether or not that gene might be associated with the mutation that causes deafness. D5S210, that is. But when you look over more and more parts of the pedigree, let's look down at how those individuals pass their genes on to the next generation. What's common about every individual that has deafness in this generation? There's one set of numbers that they inherited from Dad, say. Dad was deaf. 2 of his 3 sons were deaf. Which part of the chromosome did they inherit from their father? So let's see, 8-3, let me just circle that. 8-3-2-1. 8-3-2-1. Father had 8-3-2-1. Okay, so there's part of a chromosome that 2 sons that are both deaf inherited from their deaf father. Is it just that section?
No, it goes in
3-3, so every genotype or every haplotype that's associated, 3-3, 3-5-3, holy cow. So that son got his whole chromosome 5 from his father. Means there was not very much recombination on that chromosome. What about the other son?
only got a
Looks the same. So we've got a whole chromosome inherited from the father to 2 of his sons. So far everybody that has that chromosome gets deafness. Is that true when you look to the right? What genes can we start ruling out as causing deafness when we look at other individuals? Start from the top and work down. Right, so none of these individuals has 8. So we know that's not part of the deafness haplotype. They all have 3-2-1, but now we get some individuals that have 5, so that's not associated. So what it looks like we're doing is we're looking in that little block of genes on chromosome 5. And that's essentially what you all told me, those of you that tuned in. So some of you highlighted haplotypes that appear responsible for the disease down here, blocks in blue. And it turns out, when you zoom in on that same part of the pedigree we were just looking at, there are some individuals that only have a tiny little part of that block that are associated with deafness or not. So you look at many individuals, and you can start narrowing down smaller and smaller blocks of the chromosome that associate with deafness. We already talked about this. There are some individuals on this pedigree that were missing. This isn't actually missing a chromosome, I hope. The person probably had more issues than just deafness if they were missing a whole chromosome. So this just means that we didn't have the information as I just mentioned. So somehow one copy of their chromosome 5 we don't have the genetic information for. It doesn't mean that it wasn't in the person. We just don't have that data. So that's the process of haplotype mapping. You look at the phenotypes, you look at the genotypes, you just look for associations, correlations. And it turned out that it is indeed in this tiny little block between the genes IL9 and GRL that are most perfectly correlated with deafness. That is, when you look down here, the individuals that have 3 and 2 or something involving just 2 or just 3 all get deafness. So it turns out that when you look over the entire pedigree, that's the part of the chromosome that you can reduce to the smallest possible section of the chromosome that seems to be perfectly correlated with this trait. Except what? What's the inheritance pattern of this pedigree? Is it dominant or is it recessive? It's in every generation. It's dominant. So if having either a 3 or a 2 in the region of the chromosome that contains these genes causes deafness, what's going on here or there? These individuals have that haplotype, but they're not deaf. You just say, oh, it's incomplete penetrance.
They all have a 1 and 1. Right.
So both of these individuals have, on the other chromosome, 1s and 1s. But if this is dominant, then only having a single copy of that 3-2 should give you deafness, if we're right about that part of that chromosome causing deafness. If inheritance of 3 and 2 causes deafness, those individuals should be deaf. But they're not, for some reason. So in the real world, genetics isn't perfect. Sometimes other factors, like exposure to loud sounds, cause deafness. These individuals might have other parts of different chromosomes that give them mutations that actually prevent them from getting deafness, even though they got the bad part of chromosome 5. So in real pedigrees, it's messy. Both sigma factors, okay, so we've got sigma factor, we've got TBP. We're talking transcription. Which one's prokaryotic, which one's eukaryotic? Sigma is prokaryotic, and TBP is eukaryotic. Both bind the promoter, yes. What sequence does TBP bind? Tata binding protein binds Tata. Sigma factor, I didn't ask you to memorize. This is a good question. Is this the Shine-Dalgarno sequence that they bind? Which process is the Shine-Dalgarno sequence involved in? Translation. So TLN, translation, prokaryotics, the ribosome binds the Shine-Dalgarno sequence. I didn't tell you what the name of the sequence is that sigma factors bind in transcription. There is a sequence. What it's called and what it is not really important. What do ribosomes bind in eukaryotic translation? What was it? What do they initially bind to? So the very first thing the ribosome does in the prokaryote is the ribosome assembles on the Shine-Dalgarno sequence, and that's where transcription or translation starts. But in eukaryotes, how does the ribosome find a messenger RNA in the first place? It's the 5' cap. So you have two totally different, despite the fact that both prokaryotes and eukaryotes have ribosomes, they find RNAs to translate in totally different ways. Transcription is very similar in the sense that there's a protein in bacteria and there are proteins in humans that both bind to DNA in promoters and start transcription. But translation's a bit different. Eukaryotes have 5' caps. That's where the ribosomes in eukaryotes bind the messenger RNA to start looking for the AUG, where translation starts. In prokaryotes, it's all about the ribosome binding at the Shine-Dalgarno sequence. So there's an actual DNA sequence in prokaryotes. In eukaryotes, it's just that 7-methylguanosine cap. Others? What's on these other slides? No. No. Right. Oh, this was just the proof. This is the abstract of the paper that talked about that autosomal deafness case. This is just the proof that you did the analysis correctly. That they reached the same conclusion that many of you did when you turned in those assignments on Google Classroom, that the region between the genes IL9 and GRL were carrying this mutation that causes deafness. Okay, so does a poly A tail have any function other than protecting the messenger RNA from degradation? Maybe? I don't know. Does anybody else know the answer to that? I would look it up on Wikipedia. Honestly, if I wanted to learn more about the poly A tail, that's where I would go first. What does the poly A tail do for the messenger RNA other than protection? I'll tell you one thing, it gives researchers a way to know what's in messenger RNA. So that's not a biological purpose for adding the poly A tail to a messenger RNA. But in the laboratory, when people want to study what genes have already been transcribed, they use techniques that identify which RNAs have lots of A's in them, poly A tails. And you can actually extract that subset of messenger RNAs out of a cell. So you know what genes are being turned on and what genes are not at any point in time. So other than that, no. It's mostly protection. Do recessive traits always skip a generation? Does anybody want to answer that? No, you don't want to answer it, or no, they don't always skip a generation.
They don't always skip because the parents are both barriers and chance that the kids will be affected.
Okay, so let's assume we're talking about an autosomal recessive situation. Both parents are carriers. So this is a pretty typical case, actually. This is often where you start seeing why the recessives often skip a generation. So both parents are carriers, neither of them are affected. What's the chance that one of their kids inherits the mutation from both of their parents? So they each have a 1 in 2 chance of getting the mutation from one parent. So the probability of inheriting both of the mutations is 1 in 4. So one of these kids probably is going to have the disease. A couple are going to be carriers, and one, by chance, inherits the plus allele from both of their parents. So it's skipping a generation. It's absent, and then it's present. Could somebody describe a situation where you would see multiple generations in a row having a recessive mutation show up in a phenotype? Let's start with an affected parent and a carrier. So it's in one generation. How many kids do you want this lucky couple to have? 10. Oh, geez. Somebody pick a more reasonable. No. 4. Okay, we'll go with 4. Good grief. We're working on a time schedule here. So we've got a carrier and an affected parent. This is recessive, autosomal recessive, let's say. So we can avoid the complication of X linkage. Okay. So every kid gets the mutation from the affected father. So at least everybody's a carrier. And then how does mom's genotype? She's a carrier, so she's heterozygous. How does that pass down to her kids? 50-50. So half the kids get a plus, half the kids get an asterisk. So we have some affected individuals, and like I said, everybody else is at least a carrier. So here's a recessive disease that didn't skip a generation. And that's because mom was a carrier. Both parents have to have the mutation in order to, even if it's recessive, to pass it to the next generation. What happens if brother and sister, let's be naughty, have 4 kids? We can do this sort of thing in the classroom. So here's a recessive disorder. Does it look like it's acting in a recessive manner if you just look at this part of the pedigree? It's passing from 2 affected parents to all of their offspring. That looks kind of dominant, but it's not. It's just inbreeding, which is why you're not supposed to inbreed, because if your family happens to have those rare mutations that are recessive in them, inbreeding increases the chances that those mutations get paired together in kids, and then they show these recessive disorders. Okay. Let's say this brother marries a woman, they have 3 kids. Are there going to be any affected individuals over there? What's your prediction? We got one head shaking no. We've got a couple people saying no.
They just draw father gen.
Right, we don't know what the genotype of this, I mean, he married or didn't, whatever.
Doesn't matter.
What happened in Vegas stayed in Vegas, except for the 3 kids. So we don't know the genotype of the mom. Common assumption, and again, this is assumption, so you get to decide if you're faced with this sort of a question on an exam or in life in general as a geneticist, you have to make a decision. What's the genotype of that individual? If they're unaffected, they're either plus over plus or, like you said, they're a carrier. We don't know. So that distinction, whether or not this female is wild type, homozygous, or heterozygous, is going to dictate the phenotypes of their kids. Probability that the male in this case happened to have kids with a female who also happens to have the same mutation is probably pretty low. Probably. Who knows? So if the female's genotype happens to be homozygous wild type, some of the kids are carriers. They inherit the mutation from the dad. Maybe 2 out of 3. Should be 50%, but with 3 kids you can't have 50%. So this is something else that recessive diseases do sometimes. When you have a carrier marry somebody with a wild type genotype, it skips not just 1 generation, it skips multiple generations. You see that phenotype disappear from that lineage until the point in time when you've got inbreeding or you have a carrier happen to marry or have kids with at least. Sorry. Somebody who is a carrier by chance. And then you see that phenotype reappear. So yes, there are situations where recessive diseases don't skip generations. But that usually only happens in part of a pedigree. And you see other parts of the pedigree where it does look like it skips a generation or the phenotype disappears entirely, which would be a clue that it's recessive. If it's dominant, if one person has it, they should pass it to half of their kids. So you should not see dominant mutations just all of a sudden, poof, it's gone from that branch of the pedigree. That's a trait that's more similar to a recessive mutation.